All right, so we've talked, if you remember all the way back to chapter 12, I think, we talked about the cell cycle and how if the cell cycle was improperly regulated, then that might lead to cancer. And that's what this section is about. It's about it's about the genetic changes that can arise that will lead to cancer. So it's actually, everything comes back to the DNA. Everything comes back to how the genes are regulated, how much protein we're making, and that's what can lead to cancer. As far as, I mean, we don't know everything about cancer, but we do know a lot. So basically, so we don't do those. Okay, so cancer can arise from mutations in genes that control cell growth and division. So if we have a gene that regulates how often a cell divides, like you should divide every this often, and suddenly we have a mutation in that gene and it's hyperactive and it tells it to divide all the time, then we're going to get a tumor. There are also some things called tumor viruses which can cause cancer in animals. Okay, so there's something called oncogenes and proto-oncogenes. Oncogenes are cancer-causing genes. They basically stimulate the cell cycle to happen when it shouldn't. So if the cell is not ready to divide because there's no more need for any cells, there's no need for any more cells, or there's a damage in the cell or anything like that, the cell normally wouldn't divide, but an oncogene is going to tell it to divide. And an oncogene is a mutated form of something called a proto-oncogene. So proto-oncogenes are normal. They're in our cells. They are responsible for normal cell growth and division. Okay? So proto-oncogenes are normal. They say, okay, you're ready to divide, go ahead, divide. But they know when they should do that. But oncogenes are like permanently turned on versions of proto-oncogenes that tell the cell to divide all of the time. And so they can change back and forth. So if we have a proto-oncogene and suddenly there's a mutation in the proto-oncogene and now it's coding for a protein that's stimulating cell division all of the time, then we're going to get a tumor, which is bad, obviously. So there's 3 different ways that your cell, or that your book talks about how the genetic changes that can turn a proto-oncogene into an oncogene. So remember, proto-oncogene is normal, codes for regular cell division when it's ready to happen, and oncogenes are bad. They tell the cell to divide all the time, even if it shouldn't, which leads to a cancerous tumor, or can lead to a cancerous tumor. So one of the ways that this can happen is, here's our proto-oncogene, it's on DNA. I mean, it's a gene, it's on DNA, obviously. What can happen is if a piece of a chromosome breaks off and is translocated or transpositioned to a different, to be by a different part of DNA, it might be by a different promoter, might be by a different region that leads to more transcription of that gene. So now suddenly, because we placed it next to this new promoter due to that translocation or transposition, now that gene is going to be transcribed way more than it was in its original position, which means that we're going to get way more protein. And that protein can basically lead to the cell dividing in a number of ways. So there's not just one proto-oncogene, there's a lot of them. So that's one way that a proto-oncogene can be turned into a oncogene and stimulate cell division when it's not necessary. Another way is if something happens and suddenly there's multiple copies of this gene. So basically, there's an error, and now this gene got copied a bunch of times, and now each of them is going to be transcribed like normal, but instead of one copy being transcribed and getting the normal amount, if there's 3 copies of it, then we're going to have 3 times as many. It means there's going to be 3 times as many of this protein. It still functions totally normally, but it's just, there's too much there. There's 3 times as much of this protein than we need, so there's 3 times as many proteins telling the cell to divide. It's going to divide when it doesn't need to, which is bad. And then there could also be a point mutation, which we talked about, which could, if it was in a control element, so within a section of the DNA that controls the transcription of the gene, so if the mutation were over here, and suddenly now this control section is saying, transcribe, 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 much more than it was before, the protein is still normal, still doing its job, but again, we have too much of it, so too much of it is telling the cell to divide too often. Or the normal proto-oncogene might get a mutation actually in the gene for the proto-oncogene, and now instead of the protein being normal, it might be mutated and it might be super, super, super active. So one of these is the same as a bunch of the normal ones. It's just very, very strongly suggesting let the cell divide even when it shouldn't. So those are the three ways that it might be possible for a proto-oncogene to turn into an oncogene. Proto-oncogenes are normal, encourage cell division when the cell is ready and when it's healthy for the body or for the organism. Oncogenes encourage cell division all the time, which can lead to cancer because a cell dividing over and over and over again is obviously going to lead to a lot of cells, which, if unregulated, would be a tumor. There's the three reasons that I just said. Movement of DNA within the genome, so a section breaks off and attaches to another section and is now by a more active promoter, so there's going to be more transcription. Amplification of a proto-oncogene, which means that we just have more copies of it, so it gets transcribed more, which means there's more mRNA to attach to ribosomes to make the protein, so now we have more of the protein. Or point mutations either in the proto-oncogene itself, turning it into a hyperactive form of the protein, which acts as an oncogene, or if we have a mutation in a section of the DNA that controls the transcription of the proto-oncogene and suddenly it's being transcribed more, that would lead to it being an oncogene as well. So there's proto-oncogenes and oncogenes which encourage cell division. So a normal proto-oncogene encourages the normal amount of cell division. A mutated oncogene encourages too much cell division, which leads to a tumor. So that's one way that cancer can arise. Another way is in these things called tumor suppressor genes. So tumor suppressor genes function normally to prevent your cells from dividing too much, to prevent them from dividing when they're not ready yet, or to prevent them from dividing if the DNA is damaged, because your body knows that passing on damaged DNA is bad. So it has all these mechanisms in place to only allow cells to divide when they're ready. So this is kind of the reverse. Proto-oncogenes encourage cells to divide. Tumor suppressor genes do the opposite. And so a mutation in a tumor suppressor gene, suddenly cell division is unregulated. Cells can divide whenever they want to. Cells can divide even if they're damaged. Cells can divide even if they're not adhered to something. So normally cells need to be stuck onto something to divide. Basically, all of your cells, most of the cells inside your body are stuck in one spot, right? Blood cells circulate, obviously, lymph cells circulate, but most of the cells inside your body are stuck in one spot. And that's because they need to be, because we don't want them to be able to grow and change and divide everywhere. We want them to be where they belong. We don't want our eye cells breaking off, traveling to our lips, and suddenly growing eyes on our lips, right? We don't want that. So these tumor suppressor genes prevent division unless everything's perfect, unless they're adhered to something, unless they have space to divide, unless they're ready to divide, unless their DNA is perfect. So if there's a mutation suddenly in one of these tumor suppressor genes, now the cell can divide even if it's bad, even if it's not ready to divide or it's not in a good situation to divide. So that's the opposite. So tumor suppressor genes prevent cell division, but if there's a mutation, there's going to be cell division that is bad. And proto-oncogenes encourage cell division, but only when it's good, but an oncogene encourages cell division all the time. So basically, there's two main genes that are involved in human cancers. The RAS gene is involved in about 30% of human cancers, and the P53 gene is involved in 60% of human cancers. So, or mutated forms of these genes are. So we don't know exactly how a normal cell becomes a cancerous cell, but we know that these guys play a role in it. So RAS is a proto-oncogene, and P53 is a tumor suppressor gene. So they're good examples of both of them. So basically what RAS does is, here's a picture of it. RAS is this protein right here, and what it does is when the cell receives growth signals, meaning it's time for you to divide, or growth factors, RAS is in charge of going around the cell and turning on everything that needs to be turned on to eventually make a protein that stimulates the cell cycle. So it's a lot of different steps, but basically RAS is the beginning of a bunch of steps that lead to a protein that tells a cell to divide. Okay, so RAS is the first one in there. So, but RAS is normally only activated when growth factors are present. So they'll only do this when growth factors are present. But a mutated RAS gene suddenly doesn't need that anymore, and it's going to, is there a picture of this? Oh, right here. So now this hyperactive mutated RAS is going to tell the cell to divide. It's going to activate this pathway even if there's no growth factors. So normally a cell needs growth factors to divide, but if you have a mutated RAS, your cells can divide even if there's never any growth factors. So that means you're going to get unregulated cell growth. Your cells are going to be dividing when your body doesn't need them to be, which is bad. And then on the other end, P53 is a tumor suppressor gene. Basically, P53, your book calls it the guardian angel of your cell's DNA. Guardian angel of the genome. So basically what it means is it makes sure that, that your cell's DNA is healthy, that it, is there a list of things? No. P53 pauses the cell cycle until your DNA is good to go. So if your DNA is damaged, P53 will make sure that your cell doesn't divide until it's ready, until the DNA is fixed. And it can directly turn on genes that will go fix the DNA. So basically P53 is a tumor suppressor gene that makes sure that the cell's DNA is in good shape before division happens. So if there's a mutation in the P53 gene, now suddenly cell division can happen with damaged DNA, which is bad. If your DNA is damaged, then you're not going to have healthy cells, and they're not going to make the right proteins, and they're going to be very bad for your body, but they're going to be dividing. So P53 is really, really important, and it helps prevent from mutations being passed on. Okay, this is a great picture. Basically, either a protein can be overexpressed, so the cell cycle is overstimulated, like RAS, or it's hyperactive, so the cell cycle is overstimulated, or if a protein is absent, like we knock out, that's what it's called if it doesn't work, if we knock out P53, suddenly there's nothing regulating the cell cycle anymore, so we're not stopping it if it shouldn't divide, and then we have increased cell division. And increased cell division leads to cancer. And normally we need a bunch of mutations to actually get cancer, which is why, like a half dozen, I think is what your book says. And so that's why cancer is more likely in older individuals, because they've had more time to accumulate mutations in their cells. And normally in cancerous cells, if you look at their DNA, they have at least one oncogene, so one proto-oncogene that had a mutation and turned it into an oncogene, and multiple tumor suppressor genes that have been mutated. So the cells are being told to divide all the time, and nothing is making sure that they are not damaged before they divide. Go ahead and read about the BRCA gene and about inheritance of cancer predisposition.